Good morning. So this is a review session for the week 4 on Monday morning. So during this session, we'll have a look of the key things that you need to know in terms of this week's assignments. And this week we'll be taking the exam 1 and the lab practical 1. So let's get started. So here's a really quick outline of what I'm looking for us to do during this quick session together. And what I want to highlight here is that our main focus will be on going through a study guide that I prepared for you for the exam 1. We'll also have a look of the other elements listed here, but that's going to be our main emphasis. And before we get to that, I just want to share with you something that I think is a always motivational thing to do, to have a look, where are you standing in terms of the entire course? So after this week, the week 4, you actually will have completed almost 1/4 of the entire course. So it seems like we just got started, but actually, once you have completed that exam, once you have completed the practical, it's not quite 1/4 of the final grade, but close enough that you already have in bag. So I think that that's always positive to remember, and at least me, I would feel quite motivated by that. Another thing that I will just quickly highlight here to show why I think that this week is a great way to start with the first exam and first lab practical. If you look, these contain the exam 1 and lab practical 1, 2 chapters that we have covered. So we have covered the endocrine system, so all about hormones, and we have also looked at the blood. Later on, for the remaining exams, there's going to be typically 3 chapters per exam that we're covering. So you'll notice that this week, actually, the amount of material that you have to study and be prepared to cover is rather manageable. So I really encourage you to take that as a positive thing, and remember that really this exam gives you a great chance to jump the ground, hit it running, and really get a really good set of points for moving ahead in this course. For that reason, actually, the lab practical, because we have only covered 2 chapters, and to be quite honest, there is only so much anatomy that we can find from these 2 chapters. Lab practical is only half of the length of what the practicals will be in the future. And like I said, with the exam, only 2 chapters covered, very reasonable questions. I set you up for success for that. So let's move on. So this week, there is no new assignments that I'm asking you to do. So if you're all up to date with your coursework, with your homework, you're all set. But if you have fallen behind with any of the coursework, if there's anything that you didn't get a chance to do before the deadlines, this week is really an amazing time for you, after you've taken the exam and after you've taken the lab practical, to make sure that you are caught up with those. Of course, nothing prevents you from making sure that you're caught up with those and then taking the exam and then taking the lab practical. But I really do want to encourage you to make sure that you do not miss the exam and you do not miss the lab practical. Because if that happens, I'm really not usually as open for giving extensions for those as I am with all other work. So this is just a quick reminder that if you are asking for any work to be extended, opened, I'm totally happy to do that. All you have to do is let me know, and let me know before you're going to attempt. The reason why I'm asking you to let me know if you need it reopened, if you need an extension for that, is that if you go and complete that work and I haven't reopened, there's a very good chance that that score that you achieve on Connect does not transfer to your actual grade record on the Canvas. So what do you need to do? You need to send me a message, and I do encourage using the Canvas messaging system. That's always the quickest one. I will monitor the email as well, but I encourage using the Canvas messaging system, and make sure that you do include the details of the actual assignment that you do want me to open, because what happens often is that I receive a message that just says that, oh, I need a little bit more time for this week, or not even what week it is. And then I'm left there scratching my head that, is it all assignments? Is it one assignment? What week are we actually talking about? So try to be very specific that what assignment are you talking about? And that's just so that I can quickly process your request and we can get those reopened for you. We can get that extension in place. But what matters for me is that you do get those points. Like I said at the beginning of the course, the secret to succeeding on these online courses is really just staying on top of the time workload and making sure that you collect all those little points along the way, because even though they may seem like little points, they all add up to your final score. So what are we doing this week? Well, there's only 2 pieces of assignment that I'm asking you to do. So the first piece of assignment, and this is the piece of assignment that we will be focusing on this review session, is gonna be the exam. So exam is worth of 100 points. That gives you a rough idea that how much should you put effort on preparing for it, completing on it, and how important it is to get it done. If you look at exam 1 is worth of, like I think that all exams, worth of 100 points, and we go back here and look at the assignment points for the all other weekly assignments. Last week we actually had 2 labs, so there's a little more points available, but it really is that taking those exams carry quite a lot of points towards your final grade. I really encourage you to make sure that you get all of those completed. So we'll be focusing on the exam 1 on this session in terms of reviewing the material. And I'll mention a few words about the lab practical 1 as well. That's half of the points of the exam, so 50 points. And I think that that's fairly common in all of our lab practicals, that they're 50 points. I might be mistaken. That's just from the top of my head. So I make no guarantees. There might be changes on that, but that's the rough feeling that I do have. So this week, only these 2 assignments or 2 pieces of work that I'm asking you to complete, the exam and the lab practical. Both should be completed. However, if you've fallen behind with anything, make sure that you use this week as a chance to catch up on that work. And make sure that you contact me before completing any work that you might have missed out or not submitted on time, and you let me know details. I will then confirm that, and then you can get that done. That is just so that we make sure that you get those points, they actually get recorded. So let's have a look what I've got for you to assist you to excel on this week's content. So for the exam 1, I prepared a study guide, which you will actually find it's already been out there on this module. Same goes for the future modules. Some students even enjoy having the study guide printed while they're studying the chapters ahead of the exam. So they use that study guide to guide that week's study of the content material so that they have a fairly good idea where to focus. But it's up to you. If you haven't done that, that's absolutely fine. You have the study guide. You'll find it there on the Canvas. And I'm gonna now jump out of this screen. And what I'm wanting to do is show you exactly where to find the study guide. So let's jump to the Canvas course, and you should be seeing my view of the Canvas course. I'm gonna jump now to what you would see as a student. So you would see something like this. And when we go to this week's module, so for the week 4, you keep scrolling down, and you will see that I have here lecture study resources, study guide for the exam 1. And when you open that, that pulls up this study guide that I talked to you about. You'll notice it is a blank one. So it contains quite a bit of gaps here and there. Usually these gaps are an indication that that might be something important that I want you to know about that topic. It might be an important term, important concept, or so on and so on. So I do believe that in terms of learning styles, actually writing it down helps to make sure that it sticks to your mind. So I encourage you, since it is an open book test, to make sure that you have the study guide done and you have the gaps filled into the study guide, because pretty much every single question in the exam has been pulled from this study guide. So if I was a student, I would think that it might make sense to know the answers of these gaps as well. And you'll notice that most of the time they are very easy. There's a clue either before or after what could the answer be to these gaps. But sometimes they are also a little bit vague. That's because this is following my thinking process. So your thinking process might differ from me. So I do encourage you to try to get the answers. And probably the best way to do it is by joining me on one of these review sessions or watching one of these recordings that you might be watching now. I go through the pretty much same material in all of these review sessions. There might be a little differences based on the questions that come up, depending on who's attending to the session. But so that's the study guide. It contains really all that you need to know for the exam. You'll be also able to download it on your computer. So this is just to remind you that that is something that might help you. I'm by no means requiring you to do that. It doesn't carry any points. I'm simply putting it out there as an extra resource for you. And let's now jump back . Sorry, I'm moving from one screen to another. So we're going to jump back to the Microsoft PowerPoint presentation that I was looking with you. So I think we finished here looking at this study guide that I had made available for you. And that is going to be the main focus of today's session. We will go through answers on that study guide. Another thing, how are you going to prepare for the lab practical then? Well, a lab practical is focusing on the anatomy aspect of the course. So it's all going to be about you being shown a picture and asked to identify a structure or structures on that picture. So really identification exercises. They follow the format of the anatomy practice or practice anatomy. I don't remember which way around it was named in your study module. So you've completed the practice anatomy for the endocrine system and blood chapter. If you have completed those, you have seen what is going to come up in the practical test. So in the practical, you simply, rather than moving words or titles to the right places, you simply fill in the answer to a text box. So rather than being given a list of potential answers, now you have to remember that answer yourself. Remember again, it is an open book test, so you can use your notes. I would encourage you to make sure that you take very good notes. Take notes especially about the things that you do not know when you're practicing. The other thing, before you take the lab practical, if you feel a slightest a bit unsure about the material, I really encourage you to take this practice version of the lab practical. So what you'll end up noticing in the practice versions, and these are available for every practical that you will be completing as an exam on this course, they actually contain all of the questions that you will see. And it is worth going through those practicals, especially as we move to the later chapters where the amount of potential questions is way larger. Go through them multiple times so you've seen all possible questions that might come up in the practical exam. This is because the questions to the actual lab practical exam are pulled from a larger pool of questions. So it is a good idea to go through that entire pool by taking the lab practical practice multiple times. So make sure you get that practice done so that you feel comfortable that everything you've seen, you know, and there's no new things coming up that you wouldn't have seen before. I also have, as per request, uploaded handout of the slides for your convenience to study. Some students find that useful. They are not going to be the exact same slides that you've seen on my lecture videos. That is because those slides that you see on my lecture videos are pretty much crafted all by me. So I can't share them, but I can share your publisher's slides. They contain the same content. So you can download those handouts of those slides, and they should be available on every module. Later on the course, I think that I might be missing one or two. I will work ahead to make sure that I get those uploaded there as well. But again, if you ever miss something that you're looking and you can't find it, drop me a message and I'll make sure if it's not there, I'll re-upload. Sometimes things slide past my radar as well. But this has been done in response to a few students saying that that would help them. So before we move to there, what I want to do now, I want to move on to our study guide. So let's have a look of what do we want to know about the coming exam material. So we're going to jump out of the presentation. You might be wondering, what am I clicking away while you're not seeing it? I'm just moving from one screen to another. It's not quite as smooth as it is for students in terms of how it's laid out here. But at this point, you should be seeing a PDF file. And this PDF file is the very similar one to what you've seen before when you potentially downloaded or printed a study guide for the exam. So that might be something that you already recognize. So we will use that to cover all of the material for this exam. So study guide is what we are going to spend the majority of the time. And we're going to start with the endocrine system. Remember that endocrine system was all about hormones, how hormones are produced, how they are released, how they travel, how they act, and how they are deactivated. So that's what we're focusing on the chapter on endocrine system. So let's start with something that I try to do most of the time whenever we start a new chapter. I try to connect that to the bigger picture. So here we will see a listing of all the body systems that you have either had a look already or we will have a look later on this course. And what you'll notice that there's about 11 of those, depending a little bit on how the grouping is done. These can be sometimes combined or separated a little bit more. For example, cardiovascular is just used as a one overall term in here, whereas in our course, we'll study the blood, heart, and the blood vessels separately. But there's about 11 body systems, and endocrine system really connects with all of those. And what I mean that the hormones act on all of these systems that we will be seeing. And all of these systems have also a chance to produce or affect the production of the hormones so that they can act and affect other parts of the body. So there is endocrine system out of all the systems, in my view, is the one that's all over the place. And that's the reason why I said at the very beginning of the chapter on endocrine system that that's the one that students find sometimes a little challenging because it overlaps everything. And it overlaps also with the chapters and with the systems that we have not yet covered. So do not let that freak you out if that feels a little more challenging than something that you've seen before. And we remember that there were two communication systems, control systems in the body. And what we have discussed already earlier was the nervous system. So that's what I believe that you finished your BIO201 course. And we remember that the nervous system was very quick in a manner that it acts. So there were electrical impulses that were progressing very fast, but they also didn't last for a long time. When a nervous system sends a signal, it only stays around for a short time. So that's something that you know already from the past. But let's have a look at the endocrine system. How does that differ from the nervous system that you are already familiar with? Well, of course, the endocrine system is going to be slower. The passage of the hormones takes longer time. It can take a longer time for them to also act on the target, but they also stick around for much longer in the body. So hormones are really good for a bit of a longer term responses, longer term effects than what we typically get with the nervous system. So that's what we're focusing, and that is both the benefit and downside of the nervous, of the hormonal system, the endocrine system. So let's have a little bit of an overview of just some basic points that I really wanted to highlight here. So we remember that we use the term endocrine. To discuss the hormonal system. So if we discuss as a field of science, it's going to be endocrinology. So the field of endocrinology studies hormones, hormonal organs, all these things that we will be looking on this chapter. So we're looking at the production of those, release of those, travel of those, actions of those, deactivation of those, and interactions of the hormones as well. And we remember that hormones played a role in many functions. So we have a large list here, for example, reproduction. Well, of course, that's something that you might think of immediately, male and female hormones. Also the growth hormones. Our growth is dependent on those rises or triggers given our body and rest of the parts of our body by various hormones. These are our growth hormones. They oversee the development. Many maintenance functions, so we have right amount of electrolytes, waters, nutrients, and so on in the body is through the hormones. And again, regulation of processes, energy balance, cell metabolism, as well as the body defense mobilization. Remember, all of this is via blood. So it is through the blood that we're working with the endocrine system. Well, you might have heard in the past about the difference between different kinds of glands. We have two kinds of glands as a large groups. We have the endocrine glands, which are the topic that we are focusing on this chapter, but we also have something called exocrine glands. So what are the exocrine glands? Well, exocrine glands differ from endocrine glands in a way that they have physical ducts that carry their secretions from the gland to the target. So that gets passed from one to another by a duct. So it is a duct that carries the secretions from exocrine gland. For this reason, these secretions don't really spread around through the entire body. These secretions act very locally. And some of the good examples, just to get that kind of a concept a little bit better wrapped up in your head, would be, for example, the sweat glands or tear glands or salivary glands. These are all glands that we know. So what I've drawn here is just a rough diagram of one gland. And it has a duct that then carries the secretions from this gland to act on the target, which is located where the duct opens. So it acts very locally. It is a local process. You can think of sweat is secreted from those glands to that very spot where that duct opens. Tears from lacrimal gland opens up at the corner of the eye through an opening that we can actually see if you look real closely. I don't know if you had a chance to review the eye and nearby structures, anatomy and physiology. Same salivary glands. We'll actually have a look of those three main types and how each of those have a duct, and that where that duct opens is very specific within the oral cavity. So that's something that will come back later on this course. So that's the other kind of gland. But we are, of course, focusing on our endocrine glands. So it is going to be all about hormones. So these do not have ducts. Instead, the secretions are released to the bloodstream. And for that reason, these secretions spread throughout the entire body. And there's a bunch of different endocrine glands. Some of them we have already had a look. So pituitary gland, pineal, thyroid, adrenal gland, so on, testes, ovaries. So if we compare in terms of the diagram of what we just saw, here we might have our endocrine gland that produces a hormone. Oh, here we have hormones that this gland produces. These hormones are secreted to the bloodstream, and within the bloodstream, they spread to all parts of the body. So this can be long distances, unlike what we saw in exocrine glands. And eventually, this bloodstream carries them to the target. So you'll notice how distances would be different. And also probably in certain hormones, at least, the timeline that it takes to reach the target might be also different. But remember that our interest is, of course, on this chapter, on the endocrine glands. I just think it's a nice thing to have a basic idea of the whole concept. And one of these glands that we will discuss, that's usually the one that we love to use as an example in my field, is pancreas. This is because pancreas is actually an odd one. It is both an exocrine and endocrine. So it has functions as both of these. So the exocrine function, which we will look at when we discuss about the digestive tract and digestive system, is actually production of the pancreatic juice. This is the most complete digestive secretion that we produce in our body. So we'll have a look at that later on, further down the line on this module. What we are interested now is the endocrine function, which is all to do with the blood sugar level regulation. So regulating that the amount of blood sugar remains enough, but it's not too high. And that's done mainly through insulin and glucagon. We'll have a look of that a little later. But one good thing to note, like I said, that there are two functions for the pancreas. It works both as an exocrine and endocrine gland. Okay, what we completed as part of our study of this chapter, we completed a handout that looked something like this. That was our first week's review session. So I encourage you to make that handout available for you. If you haven't had a chance to do that, it might be a good idea to complete it as using the videos that I've recorded for you from our week 2 review session. So during those review sessions, we completed this handout. And I'm not going to go through this handout here and now in this form. Rather, you should have it handy, and we will go through each of these glands one by one, and very same topics, very same themes will come up later on this study guide, but I will have that laid out a little differently from what you've seen before. So let's have a look what we've got. We started with the hypothalamus, I believe. And the reason why we often start with the hypothalamus when we're reviewing the endocrine gland is that hypothalamus is really sometimes known as the master gland. That's because hypothalamus has kind of a control function over all other hormonal glands, all other glands of the endocrine system. So what we end up seeing, if we do a rough sketch, here we have our hypothalamus. We have this little streak that connects hypothalamus. This streak is known as infundibulum, and it connects to the pituitary gland underneath it. And we remember that pituitary gland had 2 lobes. It had both anterior and posterior lobe. So we'll have a look of the pituitary gland as the next one. But what I want you to see is that pituitary gland receives the signals from the hypothalamus. And the structure that anatomically connected these two was our infundibulum. So this is a cross-section, kind of a stylized that you're seeing, that I scribbled there. Hypothalamus then itself receives neural signals from the other areas of the brain that then trigger its actions. I think that those are the biggest things that I can think of to highlight there, just to show the passage of the messages. Okay, so I think that we're ready to move on to the pituitary gland. And what you'll notice with pituitary gland, what's important there is that we actually have 2 lobes. And that becomes very important as we are looking at the differences between these two and the hormones that each one releases. So the posterior pituitary gland, the one that we would be seeing on this diagram here, this one, posterior pituitary gland receives signals from the hypothalamus, both as a neural signals, but also in a hormonal signals. And there's 2 hormones that I want you to know from the posterior pituitary. The first one is gonna be oxytocin. What oxytocin does is that oxytocin is a hormone that plays an important role in the childbirth, so the delivery of the baby, and also overseeing the lactation. Both of these actions are actually examples of positive feedback. That was a rare one for us to see. If you remember that I highlighted that most of the time in the body, it is negative feedback that we get as an example of the feedback mechanism. But this time with the both childbirth and lactation, it is positive feedback. So if you remember our little diagram, and we will actually have that later on this study guide, but we remember that there was some kind of a stimulus, and that caused for a sensor or receptor, either word can be used, to make a note that there is now some kind of a change. So stimulus was always a change. And body responds to these changes. That's what we're interested in. So changes were detected by either a sensory or receptor organ, which passed the message to the control center. Usually this control center in humans is the brain. There are a few times when we can find, especially in other animals, that it is something else. But we're going to assume that most of the time it is the brain. And within there, we had a set point. This is a range of values that defines what that stimulus should normally be. So if we look at this as a continuous line, let's use an example of temperature. So it can be really cold. And I'm just going to use Celsius degrees there. It can be minus 30 Celsius, or it can be really warm, 60 Celsius, positive. So human body is not able to handle the body temperature going to negatives, not even close to negatives, because at negative temperatures in Celsius scale, the water freezes. So that would already burst all the cells. As the water, as it freezes, it would expand, and that would not be good. In fact, also, if the temperature rises too high, we would also have issues, because at 100 Celsius positive, water boils. And if the temperature goes even slightly higher than what is normal body temperature, that starts for various enzymes to degenerate, to break apart. And our body relies on these enzymes to work. So it is only within a very narrow area on this scale that human body temperature has been set. And it is about 37 Celsius degrees. What that is in Fahrenheit, you'll have to look up. I operate with Celsius scale because that is the international scale that we use in sciences for temperature. So my apologies that I have not studied by heart the conversions to the Fahrenheit scale or any other scale. So human body normally 37 degrees of Celsius. If it gets any colder, we actually already start having body systems slow down, eventually shutting down, and you will die. If it gets any higher, like I said, for example, enzymes get degenerated, don't stop working, and again, your body will not function anymore. So what do we do in terms of the control center? If there is a change, that change is sensed, message passed to the control center. Well, we want to act on that. So it can be effector or any other word that I might have used. That is then the actual process how we respond to these changes. And you remember that most of the time, the response in a homeostatic mechanism loop was to stop this change from happening. So I'm using this arrow that doesn't have an arrowhead, it simply ends in a line that tells us that it stops that from happening. So this would be an example of negative feedback. And most of the homeostatic control mechanism loop responses are negative feedback, stopping something from happening. Every now and then we see different from that. We might see that this effector actually makes that change to happen even more. So we're now doing positive feedback. And this is much more rare. So oxytocin was one example of that. So when we have childbirth starting, we have childbirth going, that only triggers this childbirth to happen even more. So the baby doesn't stop somewhere half through there during the delivery. Once it starts, it really is going to happen through the positive feedback mechanism. Same goes for the lactation. So the milk secretion from the breast gland, the suckling of the baby triggers more milk to be released. Well, baby is likely to suck more and more milk is released again, and so on and so on. So that would be an example of the positive feedback mechanism. So that was the first hormone from the posterior lobe of the hypothalamus, a posterior lobe of the pituitary gland that I wanted to discuss with you. So posterior lobe is controlled by the hypothalamus, both neutrally and through hormones, released oxytocin. And the second hormone that I want to loop with you is called ADH, often abbreviated with these three letters. So what does ADH stand for? It is anti. So opposite of something that we will see, and it's antiuretic. So against, opposite to urination, antidiuretic hormone. So what it does, it makes us produce less urine. So the arrow downwards is there to symbolize that less urine is produced, urinary system functions less in the presence of ADH. And we'll actually have a look at the urinary system later on this course. So that's just something to put out there. ADH function, antidiuretic hormone, to slow down the urine production process. So let's have a look at the anterior lobe of the pituitary gland. So what happens with the anterior lobe? Normally, anterior lobe of the pituitary gland receives its signals from the hypothalamus and then through the posterior pituitary, and only then these signals reach the anterior pituitary gland. So you'll notice that the posterior pituitary gland is the first one to receive these signals and then passes on to the anterior lobe if necessary. So there were a bunch of hormones, I think 1, 2, 3, 4, 5 larger groups that I wanted to discuss with you from the anterior lobe. So let's go through those quickly. So the first one that I want to discuss with you is gonna be growth hormone. So what growth hormone does, often abbreviated as GH, it stimulates the growth, so promotes the process of the growth. So for example, during your after birth, during your early life, there's a lot of growth hormone present. Baby grows in relation to its size very fast. Then as you become a child, after infancy, your growth isn't quite as fast. And once you reach your teenage years, early teens, you reach your puberty. At that point, we again that have the surge of growth hormones, and you go through the process of growth spurt. So growth hormone is responsible for all that. So let's have a look at the second hormone. The second hormone from the anterior lobe is that we're having a look of is gonna be thyroid stimulating hormone, so often again abbreviated as TSH. What it does, it controls the functions of the thyroid gland, and we'll have a look at the thyroid gland a little later in just a few moments. But notice that if there is a presence of TSH, it promotes thyroid gland to act. Well, another hormone from the anterior lobe, adrenal gland hormone. So this hormone again promotes the actions of the adrenal gland. So you'll see how these hormones often have an effect on other glands. So it's a multi-step process, and usually it's a positive effect in terms of promoting its actions. The next set of two hormones that I do want to talk about are gonna be follicle stimulating hormone and luteinizing hormone, so abbreviated as FSH or LH. Those are probably most commonly used rather than writing out the names in full. That's just simply, it makes the discussions a little bit quicker. It reduces a little bit of extra writing and so on. So we'll see especially, we don't want to do that. We'll see especially the FSH and LH when we talk about female menstrual cycle at the very end of this semester. So those two hormones are going to be very important on that, and good ones to know. So again, released from the anterior lobe of the pituitary gland. And the last one that I want to mention from the anterior pituitary gland hormone here in this context is going to be the prolactin. And prolactin, again, think of what the name tells us. Lactin refers to the lactation. And if it's pro, it's in favor of that. It promotes that. So prolactin promotes milk production and milk release. You saw that there was another hormone already earlier on, oxytocin, that played a role on that as well. So often we have multiple hormones, and it can be the interactions of these hormones that then end up affecting the final result. So these are all the hormones that we need to know, everything that we need to know about the hypothalamus and pituitary glands, two lobes that we have just looked at. Let's move on and talk about adrenal gland. What could we say about adrenal gland? Well, you remember that adrenal gland is this tiny, kind of a little bit like a pyramid-shaped structure on top of the kidneys, if you wish to describe it in that way. So if we have our kidney here, and we'll have a look at the kidneys later on this course, but they're kind of a little bit of bean-shaped structures, and one on each side. The right one slightly lower because of the liver being on top of it, taking more space from there. On top of those, we have these, and this is not to the scale I'm overemphasizing here, these pyramid-shaped adrenal glands. And these release various different hormones that might be important. And what we'll notice in terms of the adrenal gland, if we have a look of the cross-section in there of the adrenal gland, it actually has two distinct parts. We have our cortex, which is the outermost layer. So just like you saw with the brain when you studied that in your chapter of the nervous system, the cortex of the brain was that outer layer, that kind of wiggly sausage-looking layer, usually gray in color. So similarly, we have a cortex on the adrenal. And then we have inside the medulla, which is the middle part. And we're going to have a look of these two separately when we discuss about the adrenal gland. So from the cortex of the adrenal gland, there's a bunch of hormones. In fact, 25 different kinds of hormones that are secreted. And I'm going to focus on only two major groups. So the first hormone from the cortex of the adrenal gland that we're going to have a look of is the aldosterone. What aldosterone does, it has many different functions that many different physiological processes that it affects. It acts on the kidney function, in energy production, which is the ATP production, ATPase, regulating the blood pressure. The blood pressure and kidney function go hand in hand, so often it is the main function of the kidney actually to regulate the blood pressure. Concentration of the plasma is another thing that aldosterone acts on, and of course the urine production, regulating that as well. So there is a condition where you have too much aldosterone, and that's known as aldosteronism. And that would then be characterized by overaction of all of these. Kidneys work too fast, too much energy. Production is kind of amplified, rushing, blood pressure regulation, plasma concentration, urine production. All of these are on hyper. So that's our first hormone that I wanted to highlight from the adrenal cortex hormones. The second one that I want to highlight is gonna be, oh, I didn't mean to put it there. It's gonna be our glucocorticoids. So glucocorticoids is a group that contains many different hormones. So it contains hormones such as cortisol, hydrocortisone, corticosterones, so on. If any one of you has significant allergies, for example, cortisone might be something that you're familiar with, cortisone injections in more severe cases, and so on. These all play a role in metabolism, especially in stress responses and regulation of the glucose, for example. So there's a lot that glucocorticoids do. It's a large group, but as a large, if you kind of want to have an overall idea what they could do, it's all to do with the metabolism, stress responses, and glucose regulation. I think the stress responses is a really good there. So I'm going to talk about adrenaline in a second, but first I want to have a look with you about these adrenal gland hormones from the medulla of the adrenal gland. So we remember that we had two branches of the autonomic nervous system. We had the sympathetic and parasympathetic branch. And sympathetic branch was all about these fight or flight responses. So these are, for example, going to be that you are preparing to fight, or you're preparing to run away from situations, so your heart rate goes up, your pupils shrink, your breathing speeds up, your blood is directed to the muscles, you're more alert, more ready to react. Whereas the parasympathetic branch was often described as a rest and digest branch. So in that one, we're not in an immediate danger. So that's how you would feel after Thanksgiving meal. So you're breathing deep. Blood is mainly directed to the abdominal area where we're doing a lot of digestion. So catching all those nutrients, energy. Your kidneys are working. Your digestive system is working hard. Your heart rate goes down. And so on and so on. You become a little drowsier. You're not now in this alert stage. So the medulla of the adrenal gland was especially involved with the sympathetic branch on these fight or flight responses. And some hormones that relate to that I want to highlight here in terms of the medulla of the adrenal gland are going to be our epinephrine and also norepinephrine. So what these do, remember, we're talking about sympathetic branch. So it's all this getting ready to fight for your life or run for your life. So things such as an example, the heart rate going up, blood distribution is changed. Blood is now distributed not to the guts, not to for the digestion, not for the rest, but actually to the muscles to really fight or flight. So those are the medulla hormones from the medulla of adrenal gland. I wanted to mention adrenaline, just that you remember this again to this sympathetic branch. Maybe I should add this as a third one. We're kind of already addressed that there, but just so we have consistency with the numbers. So all about this fight or flight responses. So you can consider that this is kind of an extension of what we started to discuss here earlier on. Well, let's talk about next glands. Next glands that we are gonna look at are gonna be our ovaries and testes. So these are the gonads. So these are different for the males and females. Let's start with the female ovaries. So primary sex organ of the female are ovaries, and what they do, two especially important hormones that I want us to notice from there is estrogen and progesterone. Remember that the only function of the ovaries is not the production of the egg cell. In addition to that, just as important, or if not more important function, is the production of the sex hormones. So these are the two sex hormones that I want us to focus on the ovaries. And on those ones, clinical importance is such as we use estrogen, progesterone in a lot of drug research and so on. For the males, testes. Well, what do testes produce? Testes do produce the sperm cells, spermatozoa, but in addition to that, they do produce the male sex hormone, the testosterone. So that's one to notice for the males. So I'm adding that there. So those are gonadal hormones, primary sex organ hormones. Let's move on to the kidneys and have a look of what do we find with the hormones from the kidneys. So the hormone released from the kidneys is known as renin. And we'll talk about, when we talk about the urinary system, of renin, angiotensin mechanism and so on. But what renin does, what the kidneys do, is, I keep highlighting this function. Of course, they have other functions as well, such as production of the urine. But the important one that I want to highlight here is the blood pressure regulation by kidneys through renin. So if you think why the word renin, that doesn't sound like a kidney. When we talk about kidney sciences, we talk about renal sciences. So you don't have a kidney doctor, you have a renal doctor, and so on. So that's just, you don't do kidney imaging. You don't take images of kidneys through ultrasound or MR or so on. You get renal imaging done. So that's just kind of to expand on that term, where it might come from. The next one that I want to talk, we're going to come back to leptin, but I want to talk about the pineal gland. Pineal gland is this tiny, kind of a pea-shaped gland located deep within the brain, between the two hemispheres, just off the third ventricle. So if we have here a cross section of the brain, we can actually see it when we dissect the sheep brain. I have quite a nice dissection video of the sheep brain there earlier on in your learning modules, I think on the second week. That's not something that I'm going to examine you on. But if you're curious to see the pineal gland, I do show it there from the sheep. So what the pineal gland does, what the hormone associated with the pineal gland is the hormone known as melatonin. It is not melanin. Melanin is something totally different. Melatonin is what we're focusing on, that the pineal gland secretes. And melatonin has all to do with the sleep-wake cycle. So for example, people who have trouble sleeping, they might have to resort to melatonin supplements. People who travel a lot of between time zones, they might need to resort on melatonin drugs as well in terms of adjusting their sleeping pattern. So that's the pineal gland. I don't know, should I even number those when it's just a single hormone that I'm highlighting there? Well, for the sake of consistency, we'll keep doing. So let's move on to the next one. We're going to have a look of the pancreas. And I already mentioned pancreas earlier on. That was the one that we really digged as anatomist, physiologist, because it is kind of an odd one. So let's have a look of the pancreas. Where is pancreas located? Pancreas is within your abdomen, kind of this ice cream cone shaped structure, just wrapping to the inside of the first part of the small intestines, duodenum after the stomach. And you'll learn all about the pancreas later on when we go through the digestive system, but our focus with the pancreas is going to be the endocrine function, so the production of the blood sugar regulating hormones. And there's two hormones that we're going to look from the pancreas in its endocrine function. These hormones are going to be insulin and glucagon. So these two hormones play major role in regulating the blood sugar levels. And you know, some people end up developing diabetes or are born with type 1 diabetes. So type 1 diabetes is the one that you're born with, in that your body doesn't produce these hormones, or type 2 is then late onset, more of a lifestyle dependent issue with these. So it can be managed a little bit better, the type 2, by changes in lifestyle and so on. Type 1 diabetes patients usually end up having to take insulin injections for all of their life. Let's have a look at the parathyroid gland. So we talked about, did we talk about thyroid gland? Not yet. We'll have a look of the thyroid gland a little bit later. Yes, it's coming up. Oh, now I know, I alphabetized this. So actually, let's go through the thyroid gland first and then come to parathyroid. So where do you find thyroid gland? You find thyroid gland on your neck, on the anterior surface of the neck, so on inferior to the larynx. And if we try to describe what does a thyroid gland look, it's sometimes described as having this butterfly shape. I think that that's a little bit of a creative way of seeing it, but it does have two distinct lobules that are then connected together. So that's kind of where the butterfly shape description of the thyroid gland comes from. Well, thyroid gland produces hormones T3, and that then gets converted to T4. These hormones are not really what I want your focus to be drawn on. I want your focus to be drawn on the action of these hormones. These hormones are all to do with metabolism. So if we have individuals with thyroid hormone issues, we often see issues with the metabolism. So you might have overly active. So in that case, thyroid. So in that case, you don't gain weight even if you eat a lot. Your metabolism is running on overboard and so on. Underactive thyroid, then again, even if you really don't eat a lot, you end up gaining that weight and so on. Your metabolism is very, very slow. Of course, this is not always the case with the weight gain and so on. So often I get patients who say that, oh, it must be my thyroid, I'm gaining weight. That's not always the case. So just putting it out there. We already discussed about over or underactive thyroid. Some diseases such as Graves' disease or edema, especially behind the eye, and so on, are associated there as well. And I leave it to you, if you're interested on those, to go back to the notes and have a look of those. But what I do want to discuss is to discuss about these parathyroid glands that we almost already started with. So let's jump back there. And parathyroid glands are found within the thyroid gland. They're as little dots, usually kind of a yellowish brown in color. What parathyroid glands do, they produce parathyroid hormone. And parathyroid hormone is quite an important one because of its role in calcium homeostasis. And we'll end up noticing that calcium really plays a role in many processes all around the body. So calcium is a super important one for us to be aware of, especially some of the examples that I've highlighted there, kidney function, urine production, in terms of intestines, digestive processes. Calcium, not surprisingly, dealing with the skeleton, bone strength, vitamin D activation. The list is long. Calcium is everywhere in the body. So parathyroid hormone regulating the calcium homeostasis. So an important one to know in that sense. Ooh, I was going to draw something there. So these parathyroid glands, these little dots on the thyroid. Let's have a look at the thymus as the next one. So thymus is a little bit of a tricky one in my view, because thymus is one of these things that actually gets smaller. Oh, I didn't mean to change the color of that. Gets smaller over the age. So it is easy to find a thymus in newborn or younger individual, but as you get older, thymus actually gets replaced by the connective tissue and adipose tissue. So it becomes harder to find. It doesn't mean that thymus wouldn't be working anymore. Just anatomically, it's not as present. And thymus is going to be very important in terms of the maturation side of the T cells. And we'll talk about T cells when we discuss about the immune system. So we'll discuss that at that time. But that's the site of the T cell maturation. We can go back and just quickly address the one hormone that I did not talk with you guys yet, which was our leptin. And the reason why I didn't talk about leptin, I kind of left it as the last one. Ooh, I didn't mean to change my highlights there. Yeah, I left it as the last one is because it's hard to point out a specific location for it in terms of this diagram that we saw earlier on. Instead, leptin is released from all fat cells, so adipose tissue. And what it does, it controls, it is a hormone, so released from the adipose tissues, fat tissues. And what it does, it controls your appetite. So we can have, for example, we've done this with lab experiments. We can have rats who we've either removed the production of the leptin, or we've removed leptin receptors in their body. And these rats don't have any control over their appetite. They just keep excessively eating up to the point that they become sick. They can even die out. So what we have found, not again too common, but it is a condition that we have found that some, especially younger individuals who have insane, abnormal weight gain, who cannot regulate their appetite at all, they might have issues with leptin production. So when these individuals have started getting leptin injections, they have actually resumed back to the normal body size. So another important hormone there. And those are the main hormones that we were supposed to have a look. Looking back on this diagram, I think we've covered everything from there, just in a little bit different way from what we had a look at before. And I know that in our review session of the hormonal system, we did not cover the remaining of this chapter. It doesn't mean that the remaining of the chapter would not be important or that it wouldn't be covered as part of the material for this exam. So I do encourage you, regardless, still take a little bit of time to make sure that you are comfortable with the remaining of this chapter. So let's do that quickly together. Remember, this is just a review, full story in the lecture videos. So let's talk about hormones. What are hormones? Often it's a good thing to kind of give a definition to these things. So hormones are these chemical signals. So they are chemicals. We talked about them being signals, but being a long-term, slower-acting signals. That's because they travel in blood. They can also travel in lymph. So lymphatic system is something we'll have a look, kind of a complementary system to the cardiovascular system, picking up what kind of leaks out of the cardiovascular system from the blood vessels. So almost like a drainage of the house, if you wish. So hormones are the topic that we've been looking at. And there's two groups of hormones that I highlighted. I highlighted autocrines and paracrines. So autocrines were these hormones. Here we have a cell that were released from the hormone and released from the cell and acted on the same cell. It doesn't have to be that they're released outside the cell. It can be also within the cell. But the site of action is the same cell. So that's word of knowing. Paracrines, they are then hormones that are released from a cell and travels to somewhere within the same area to another cell. So they don't travel throughout the entire, or they travel throughout the entire body, but this is not in a totally different part of the body, within the same area, but not the same cell. So I would know the autocrines and paracrines, the difference between those two. Let's have a look at the chemistry of the hormones. We can talk a little bit about their chemistry. We have amino acid based hormones. These are amino acid derivatives, peptides, proteins, steroids. Steroids are then synthesized from the cholesterol. So some amount of cholesterol is needed to make hormones, but your body will produce all the cholesterol that you need. You do not need to get extra cholesterol from your diet. And we are talking whether eoconosides is gonna be a third group in terms of chemical classification of hormones. We're still kind of debating whether it is a hormone or paracrine. So I'm just putting it out there kind of as something that we are not totally sure of. And this is the part that I really do enjoy of sciences, and I hope you find exciting that we don't have answers to everything just yet. Science is a constantly evolving field. Let's have a look at the action of the hormones. Hormones traveled everywhere. However, a good question in my mind was that, okay, or my mind would be that, okay, if we have these hormones that travel everywhere in the body through the cardiovascular system, through the circulation, why isn't every single cell in body then affected by these, since they are not locally acting, they spread everywhere? Well, this is because of these structures known as receptors. Receptors are structures on a cell surface that work almost like a key and lock mechanism for these hormones that travel. Only if the receptor and a hormone are a match, then this receptor will trigger further changes within the cell. But a wrong hormone cannot act on a wrong receptor. So that's how we choose which cells are affected by certain hormones. These cells that should be affected have the receptors for those hormones. So not all cells have all kinds of receptors. And this is very, very specific. So like I said, the key and lock mechanism in that sense, if that helps you to remember it. So I already mentioned that what the receptors did when a right hormone binds to a receptor, that then activates or can change in some way a pathway within the target cell. And there's many things that can happen there. Okay, let's have a look at a little bit of the chemistry. We have both water-soluble hormones and lipid-soluble hormones. Water-soluble hormones, since our cell membrane, as you might remember from our basic biology courses, our cell membrane, the plasma membrane, phospholipid plasma membrane, contains lipids. Water-soluble hormones are not able to pass through that membrane. So water-soluble hormones have to act on the plasma membrane. They cannot enter into the cell directly. So to have an effect on a cell, water-soluble hormone must bind to a receptor and then have a second messenger inside the cell that then passes this action further. Whereas the lipid-soluble hormones, they are able to pass directly inside the cell and act there on whatever their action is. So they can, for example, change the genes, activate genes, change cell activity directly, whereas the water solubles were not. They needed a second messenger. I have a nice list that I think I had in your lecture notes of just kind of examples about the lipid-soluble, water-soluble hormones. You can have a look of that on your own in a little bit more detail. I provided it there as well. And we discussed the concept of homeostasis already earlier on. I mentioned that that's a very, very, very important concept. So I am expecting that you are okay with that. We already talked about that. If you are not, I really, really do encourage you to go back and have a look of that. I want to remind you, homeostasis was... Oh, let's change the color. Was all about keeping the body's internal environment consistent. So maintenance of the consistent internal environment. So because we've already looked at this, I'm going to go through it really, really fast. Stimulus, some sort of change taking place that is detected by a sensor or a receptor. Message passed to the control center, which was usually our brain. Within the control center, there was a set point, the acceptable range of values, only within which we want to stay. We don't want to deviate from that. And if we are heading too far off from that, well, then the effector kicks in to stop the stimulus from happening. And you remember this was an example of negative feedback. There were only few occasions where actually we see positive feedback, where stimulus happening triggers this chain of events, where then we promote that stimulus to happen more. Examples of that I can think of: childbirth, clotting, scab formation. There's one more, but I can't remember, and that's, you don't need to know that. So there's only a few examples of the positive feedback. Lactation could be also one. I'm still certain that there is a third one that I haven't looked at, but these are the examples that I'm asking you to remember. So know the difference between positive and negative feedback. I think that the homeostasis should be something you're familiar with. If you're not, I really encourage you to look at, not just for this exam. It is a concept that I will be using as a way of explaining many other processes in our body. And hormone then can trigger further down reactions, either with other hormones. So we've already talked about these chains a little bit, or it can also trigger a nervous system responses. So these are not separate, the endocrine and nervous system, even though they work differently. There's also times when they overlap, affect each other. So I think that we have just a few more concepts to highlight with the hormonal system. Let's look at the target cell specificity. So we remember that not all receptor or cells responded to all hormones. We needed to have these receptors, and there's 3 factors that play an important role in how do we get or what kind of a response do we get for a specific hormone. So let's have a look of those. I'm just trying to be consistent with the colors in case you're wondering. So 3 factors that play a role is that how much we have of that hormone in the blood. The second factor is that how much receptors we have for that hormone on the target cells. And also how tightly the hormone and receptor will bind together. So those 3 factors all play a role in what sort of an effect that hormone is gonna do. So change in one will have a change in entire complete effectiveness of that. We can control the amount of hormone present. And that will have the amount of hormone present can have an effect on the amount of receptors. So let's think of a situation where we have very little hormone present. Well, we want that hormone still to act. So because there's only so much of that hormone present, how can we get better response? Well, let's add more receptors. So that would be body's physiological response to low hormone levels, and that is known as upregulation. Downregulation is the opposite of that. When we have a lot of hormone present, we don't want to go crazy with the response that we're getting from that, so we are slowly starting to reduce the amount of the receptors. So up or down regulation of the receptors refer to the amount of hormone present and how that will then affect the receptor. Hormone activity, a couple of things that I've highlighted there. Hormones can be circulating either free in the blood or bound to some sort of carriers, plasma components, and so on. Concentration of hormones really depends on the balance between how much of it we release and how much of it we remove. Or if we don't remove, we can just simply inactivate. And we try to have these in balance so that the hormone amount would stay in balance. If we change one, then that will have an effect. An important concept, half-life of a hormone. That's the time that's required for that hormone level to decrease by 50% in the body. So all hormones eventually get removed. They eventually either get inactivated immediately and then removed, or they might be removed directly. And when half of that has been removed or decreases, that's the half-life of that hormone, a measure for that. Also, I mentioned very early on, I didn't go into great detail about that hormones do act with other hormones as well. So there's interactions between various hormones. And there's 3 kinds that I do want to mention. I could, in an exam, ask you to be able to recognize these. I probably wouldn't go into great detail, but it would be a good idea to know these in general. So permissiveness is when we have one hormone that then requires another hormone to be present so that this initial hormone can act. So it needs almost like a permission from this other hormone so it can do its job. Synergism refers to having one hormone present, then amplifying the one hormone present, amplifying the initial hormone's effects. It's like adding a boost on that. And antagonism is, you remember in good stories, there's an antagonist. So it's opposite of something that's a bad guy. So antagonism is when this presence of another hormone stops this initial hormone from acting or kind of reduces its effectiveness. So I would know these three types of interactions that we can have. And you remember from those definitions, they are very different kinds of effects that how they can affect on other hormones. Well, I think that covers everything that we need to know about hormones. I know that was quite a lot. So I'm going to take a really quick break and I'm going to come back and discuss with you about the blood chapter, what you need to know about that. And we'll wrap up this review session. So let's pause for a second. Okay, and we're back to continue our review session. We're going through the study guide for the exam 1. And we're going to continue by having a look of the chapter on the blood. So what I want to start with by highlighting here is that on this chapter, or this chapter is the first part of a series of 3 chapters in total that covers the cardiovascular system. So we start with the blood, which is really the media or the liquid that transports material within the cardiovascular system. And then in the future chapters after the exam, we'll have a look of the heart, which is the pump driving the movement of this blood. And we also have a look of this tubing, so blood vessels, where this blood moves within. What I do want to highlight about the blood, that remember we had 4 main types of tissues, and we noticed that the blood was connective tissue. So that's the type of tissue as a large that we classify the blood as a special type of connective tissue. It connects body parts, although it's not so much of a concrete connection as in tendons and ligaments, bones. It is a liquidy connection. So blood can be described often as an internal transport system within the body. Okay, where is blood made of? Well, blood is made of in the bone marrow, and the process for production of the blood was hematopoiesis. Remember the terms. Hemato referred to the blood, and poiesis was always making of something. So when we talk about general production of the blood, that's hematopoiesis, we'll discuss specific blood type cell productions. And you'll notice that the term poiesis keeps sticking around and the initial part keeps changing. There's about 5 liters or so of the blood in an average person. That's about 8% of your body weight. Why would you need to know about blood if you're planning to move on to the field of nursing or other health careers? Well, probably drawing blood and analyzing blood content is one of the most common clinical tests that we do. I read this study some time ago that about 70% of the decisions that medical doctors do in care of their patients rely on the blood test results, blood work results. So that's very, very common, drawing the blood and analyzing it. And drawing the blood is a great skill to have. I'm sure that all of us have had blood drawn at some point or another. Let's have a look at the functions of the blood. There's many different kinds. For our purposes, I have classified them into three main groups. And you remember that I highlighted especially the transport role of the blood as a transport media, moving materials around in the body. Examples of that included, for example, transporting the gases, oxygen, carbon dioxide, transporting nutrients, waste products of the cellular metabolism, transporting hormones like we just saw. So a lot of things get transported by the blood. Well, the other functions that I have also included as a bigger groups are regulatory functions. So regulating, for example, fluid distribution in the body, heat can be transported with the blood, with the liquids in general, regulating the body pH values, and so on. Also, we're going to be having a look of the immune system later on this course. So protective functions of the blood related that, same as with the clotting of the blood, stopping a bleeding, formation of the scab. So those were the three functions of the blood. What is blood made of? Well, there's a bunch of things that we need to have a look at. And I think that we're going to start by highlighting that how do we get the blood separated to its different components. So you remember we drew the blood sample from the patient. So usually that's collected in a test tube. And then the test tube gets put into a centrifuge that spins that. And because different components of the blood have a different weight, the different components of the blood separate within this test tube into different layers. And what we're going to do in this chapter, we're going to go through all of these different layers. What do they have? What do they do? And so on. So most of the blood, content of the blood, is gonna be plasma. About, I'd like to say, just slightly over half of the blood, so about 55% or so of the entire volume of the blood is gonna be plasma. Plasma is this yellowish or straw colored, kind of a somewhat transparent, quite transparent layer or a transparent structure. It is a little stickier than what you would have, for example, just for water. So it is not quite as liquidy. What do we have in plasma? We have many different components. The proteins were the big ones that I mentioned there. In terms of the proteins, albumin, globulin, fibrinogens, and so on. The plasma does also carry nutrients, gases, hormones, waste products, inorganic ions. So when could you use plasma? Well, other than just being the matrix where all of the other cells that we'll have a look are kind of within, in addition to that, plasma is useful in clinical practice because we don't have an issue with the donor-recipient issues as we might have with the cells sometimes, especially red blood cells. Plasma can be given from one patient to another. We don't need to do matching on that. So we do see plasma being given when there's a sudden loss of a significant amount of blood volume. So we want to just get that up and then deal with later on the other things. Let's have a look of, so that was the only liquid part. That was where everything else was dissolved in. Let's have a look of the remaining parts of the blood. And we're gonna, all of these remaining parts, since they are very concrete, they are known as formed elements. Let's have a look of through the different kinds of formed elements that we have within the blood. Sorry about that. I received a phone call, so that's the only reason why I paused for a second. But we were having a look of the formed elements. So the concrete more of a cells that we can separate, or parts of the cells from the plasma in a blood sample. And the first one that we will have a look is gonna be our red blood cells. These often get abbreviated by me and others as RBC or RBCs. The proper term for the red blood cells is gonna be erythrocytes. That's there. I just slightly drew over it. So erythrocytes are red blood cells, and it's about 45% of the volume of the blood sample or blood that we would have red blood cells. So slightly less than the plasma, but still quite a bit. And the production of the red blood cells is gonna be erythropoiesis. You remember the poiesis was the making of something, and now since we're talking about red blood cells, it's gonna be erythropoiesis. Well, what does a red blood cell look like? It looks like a flattened ball, if you wish, or a flattened disc that has a slight depression in the middle. So almost like a donut that hasn't been fully pinched through, if you wish. The term that we use to describe that as being a little bit more scientific is that it's a biconcave disc. So if you take a cross section of a red blood cell, it would look something like this. On both sides, concaved in, so biconcaved. So this would be a cross section of what we just saw. Ooh, I didn't mean to do that. What we see in here, if I cut a section through this red blood cell, this is what we'll end up seeing. An interesting thing with the red blood cells that we end up seeing is that they are what we describe as being anucleate. This means that they do not have a nucleus. Well, that might kind of throw you off a little bit, because I've told you before, or I'm sure someone has told you before, that all cells have a nucleus. Well, red blood cells do start with a nucleus, but that then over the development gets removed. It's not present any longer. And if you think of the other thing that's interesting, there's also no other cell organelles within the red blood cell. And you start wondering, why would this be the case? I think I've highlighted here mitochondria as an example of that. What would happen if we had mitochondria within the cell? Well, red blood cells. Well, red blood cells' function is to transport material, transport oxygen, for example. Well, if we had a lot of cell organelles, it would be very likely that during this transport process, the red blood cell would actually use all of these resources that it's supposed to transport. So for that reason, we don't have other cell organelles present in the red blood cell. Well, if we have a look at the structure of the red blood cell, we'll end up noticing that there is actually 4 hemoglobin subunits within each red blood cells. And each of these hemoglobin subunits can carry 1 oxygen molecule. So that allows us to carry majority of the oxygen then, thanks to those. Sometimes we have that oxygen bound to those hemoglobin subunits, and sometimes we do not. Well, that has an effect on what color the blood will appear as because of the red blood cells, either concentration or lack of concentration of the oxygen in them. If you have a lot of oxygen bound to the red blood cell, it appears bright red. So often we see on diagrams these arrows that are either red or blue, and that tells when we're talking about the circulation of the blood, whether there's oxygen bound to that blood or not. So if there is a lot of oxygen in the blood, for example, these red blood cells this blood with the red blood cells within it come from the lungs, where it's been reoxygenated, and these appear bright red in color. The term for this is that we talk about oxyhemoglobin. These hemoglobins carry oxygen. If instead we do not have a lot of oxygen, if this red blood cell has given off most of the oxygen, we describe these as deoxyhemoglobin. So these have a low amount of oxygen present. They appear more of a purplish in color. We tend to over-exaggerate the color difference and sometimes talk about that they would be blue in color, but no, they don't. The deoxyhemoglobin and deoxygenated blood is really not blue, but it has that darkerish, bluish, more purplish shade in it. And this sort of a blood typically does not come from the lungs, but it comes from all other parts of the body, from the tissues where this oxygen has been given up to these tissues for their cellular metabolism. Well, why does this hemoglobin matter in the first place? Well, we remember that we needed to have these hemoglobin subunits that then carried the oxygen. Well, there are situations where something else can bind into these hemoglobin subunits, often abbreviated as HB, capital H, lowercase b. For example, carbon monoxide, which we can find in if a fire hasn't been put out properly, so for example, or a furnace hasn't been put out properly, or even in cigarette smoke, there's some amount of carbon monoxide. That actually can bind in to these hemoglobin subunits, and by binding into these hemoglobin subunits, they block oxygen molecules from binding into those. Those spaces have already been taken. So in this case, less oxygen is carried to the body, and in a worst case, not enough oxygen is carried to the rest of the body, and there might be a death or tissue death or complete death taking place. So that's why we need to know the subunit level of red blood cells. So those were our erythrocytes. Let's have a look at what else we can find. But before we do, I want to mention we can have, we usually try to aim into a situation where we have same amount of red blood cells produced as there is destroyed. So having that imbalance ensures that we have a constant right amount of red blood cells present. There is a situation where you might have too little red blood cells. That would be hypoxia. If you have too many red blood cells, actually the viscosity of the blood is changed. What controls the production of the red blood cells? Well, that's through a hormone called EPO, erythropoietin. Presence of erythropoietin increases red blood cell production. And I think we talked in our review session, for example, high altitude training, which then triggered that in a high altitude, we have less oxygen present. So there's not as much oxygen present in the blood as well, and that triggers the production of erythropoietin. That's why many athletes go and train in the high altitude camps before important competitions and so on. That is allowed, but some athletes also get EPO injected into them, which would be technically cheating. That would be doping. The logic with that and the high altitude training is as well that by getting a lot of EPO, we get then a lot of red blood cells. And if the red blood cell amount increases, the idea is that, well, then this athlete is able to transport more oxygen in their blood, so their muscles would be able to work harder because they get more oxygen than their competition. You can have a little bit of both positive or negative effects on the amount of the red blood cells, also through diet. So you need to get some iron from your diet as well. Red blood cells stick around for about 120 days, so varying a little bit of that. Once we are done with the red blood cells, though, we reuse the components that make up a red blood cell, the heme group, the iron and globins, to build new red blood cells. There's a few different kinds of disorders of the red blood cells. Anemia referred to a very low amounts of oxygen in blood, and signs and symptoms. Anemic people typically tend to be palish, tired, having breathing problems, feeling cold all the time, and so on. What can cause anemia? Well, you might have lost a lot of blood, or your red blood cell production is down, or alternatively, we destroy too much red blood cells. One specific type of anemia that I talked with you guys was the sickle cell anemia. In sickle cell anemia, we don't have these nice round red blood cells by concave discs, but because of a single base difference in the DNA, that causes one amino acid difference in the resulting protein, and the red blood cell appears sickled, kind of a crescent moon shaped. Sickled red blood cell has two significant issues that can cause, first of all, these sickled red blood cells to get stuck in places where normally nice, rounded, smoother red blood cell wouldn't get stuck. And in addition, less oxygen is carried by the sickled red blood cells. You might think that, well, then it's a really bad thing to have a sickle cell disease. And yes, it's not ideal. However, if you are able to kind of manage that you don't go crazy with the oxygen needs and so on, people with sickle cell anemia can have a just fine life. That's just something to take into account. Interesting thing that we find with the sickle cell anemia is that it is most common in the sub-Saharan Africa. So the parts of the Africa that are below the Saharan desert. So individuals from there, or having family relations, family origin from there, and having that blood line continuing from there, are more likely to have sickle cell anemia. You might wonder why on that area. Well, an interesting thing that we discussed as part of our lecture material, we noticed that there was an overlay of the map with where the sickle cell anemia is common into Africa, and also where malaria is common. So this overlay is probably not by an accident. What we end up finding is that malaria parasite, as part of its life cycle, enters to your red blood cell, multiplies in numbers, and then kills the red blood cell, and breaks and spreads further. Well, malaria is not a great thing to have. What we can do is we can have various different kinds of malaria medications. There are certain adaptations that we do to life, sleeping under nets and so on. But individuals who have sickle cell anemia, turns out these malaria parasites are not able to enter to these red blood cells. So having a sickle cell disease might be actually protective against the malaria. So there is a link between those two, and you could theorize that nothing happens in nature by accident if you believe in evolution. And in my view, it's not a matter of belief, but I respect that people might have different views. But in this course, we do have to accept that evolution happens. You don't have to think so yourself, but we assume on a scientific approach that evolution takes place. So maybe evolution has just been smarter than us and protected us from mankind in the areas where malaria is common, by giving us a disease that actually has a protective function. So evolution makes no mistakes in that sense. So this is an important topic, this sickle cell anemia. And if you watch the last lecture video review session, and I think I've even mentioned that in our announcements. If I haven't, well, it would be a good idea to watch these videos. Sickle cell anemia, what it is, what causes it, what sort of effects it has, and how is that related to malaria. That is extra credit question in your exam 1. So it might be a good idea to feel comfortable with how to explain all the things that we just discussed about. Extra credit questions are not necessary for you to answer in an exam. They are just a way to get a bunch of extra points towards your final exam score. So I want to throw it out there. Some students prefer to prepare their extra credit question answer ahead of the time in a notepad or Word or other text editing program, and then just copy paste it while you're taking the exam to the extra credit answer. That's up to you. It doesn't need to be a long answer about a paragraph, maybe 4 or 5 sentences, whatever works for you. But those are kind of roughly the questions that I'm asking in that extra credit question, if that's something that you want to kind of prepare or tackle or at least revise for. What else? We should next talk about white blood cells. And white blood cells are called leukocytes. So let's go back to our diagram that we saw earlier on. And notice the white blood cells there. There's not a lot of white blood cells in the entire blood volume. White blood cells make about less than 1% of the entire blood composition. And it usually, white blood cells collect after this centrifuging, spinning the blood sample into this middle layer between the plasma and red blood cells, between the plasma and erythrocytes, which is known as a buffy coat. It is whitish in color. So that's where we end up finding our white blood cells. Why do we have white blood cells? They mainly play a role in defense processes, defending us against different diseases. And I do want to highlight that when we talk about white blood cells, since they are known as leukocytes and production of the white blood cells, this time we're going to talk about leukopoiesis. So you'll notice how these terms do matter. So the scientific term, leukocytes for the white blood cells. And there's 5 types of white blood cells that I do want you to know. And we're going to go through them really quickly. There's a more complete description of those in your lecture videos. Remember, we had neutrophils, which are the most common type of white blood cells. They had multiple lobes of the nucleus that stained very dark. Cytoplasm was granule, and they played a role in immune defenses. The second most common was eosinophils. They are binucleates, so 2 lobules of the nucleus that again stains dark. And the cytoplasm had granules. They work largely against parasites involved in allergies as well. The third most common white blood cell was basophils, a binucleate staining dark, cytoplasm having granules. They are involved especially in inflammatory responses, allergies, so many antihistamines work through these. The second least common one was, sorry, I'm going wrong about the numbers. So most common was neutrophils. Second most common was lymphocytes. So let's just correct that there. Neutrophils, most common. Second most common, lymphocytes. And they were more of a larger, spherical in shape. Their nucleus filled most of the cell cytoplasm, no granules, so we call them agranulocytes. They are involved with immune responses, producing antibodies. Maturation from thymus is for T cells, and from bone marrow for B cells. So that's the second most common. Third most common is gonna be our monocytes. Monocytes are more, their nucleus is more of a kidney shaped, if you wish. There's no granules, so they're agranulocytes in the cytoplasm. They are phagocytotic, so they eat the pathogens that shouldn't be an unwanted material in the cell or in the body. They often are eating away, removing the pathogens. The second least common, so apologies for that earlier, was our eosinophils that we already had a look of, and the least common white blood cell was our basophils. We also discussed those as well. Why does it matter that how common each one it is? We do want to know which white blood cells you should see a lot, which white blood cells you should see a less. And you notice that I listed the white blood cells based on how common they are there. That would make a very good exam question to give you the list of white blood cell types and have you to list them in the order of how common they are. Well, I've given you a mnemonic that was never let monkeys eat bananas. So that would be a good mnemonic for remembering how common each white blood cell type is. Here I had it already, so you get to look at it twice on this review cycle. Maybe it's a review guide. Maybe that's a good thing that highlights how important they are. Clinical relevance of white blood cells. If you have too much white blood cells produced, that can be a sign of the leukemia. And too little white blood cells, leukopenia. Too little white blood cells, so few clinical conditions there to notice. And the last formed element of the blood that we have not had a look, again, like white blood cells, in very small amounts, less than 1% of the total volume, is gonna be platelets. And platelets can be found also on this buffy coat that I mentioned earlier on. And platelets actually are not full cells. They're rather fragments of cells. But they are important. They play a role in the clotting of the blood and survive only about 10 days in the body. So there's a constant reproduction cycle of the platelets. So that's as little as I'm asking you to know about the platelets. The final concept with the blood that we needed to look at was the blood typing. And we remember you needed to have a match between the donor and the recipient of the blood types, or blood types of the donor and recipient if you're doing a blood transfusion. I'm just looking for a color that we haven't used before to keep this a little bit more organized. So donor-recipient match is vital because if you transfuse blood and it's not a match, that actually will be lethal for the patient. So I went through this in quite a good detail, I believe, in one of the lecture videos. We used donuts as an example of that who can give blood to who. Let's take an example of now we have an individual with blood type A. That individual, since they're blood type A, their red blood cell surface have anti-B antibodies. So that means that they can receive blood from another type A individual, but if they receive any blood from someone with B antibodies, we will then have a clotting mechanism which will be a problem for that individual. Also, they can receive blood from O-type individuals because these do not carry any B. For blood type B, these have anti-A antibodies. So if there's any A's, whether it's from a donor A with A blood type or donor with AB blood type, there will be a clotting process, and that's not good. They can receive blood from another B type B individual and also from type O individual. How about an individual whose blood type is AB? They don't have any antibodies on the surface of the cell, so for that reason, they can receive blood from all donors. Individuals who are blood type O, they have both anti-A and anti-B antibodies on the cell surface, so they cannot receive blood from anyone who would carry A or B. So the only ones they can receive blood is from another O individual. For this reason, individuals with blood type AB are considered as universal recipients. They can receive blood I didn't mean to do that. Apologies. Let's see if I can change it back. That seems better. So individuals with blood type AB, they can receive blood from all donors. So they're universal recipients. Let's have a look at the individuals with blood type O. Well, they can donate blood to anyone. Their blood can be accepted by anyone at all. So that's why they're considered as universal donors. We also talk about individuals having a blood type that's positive or negative for Rh factor. And I didn't go into great detail of that, but what we need to know is that this becomes mainly a problem if the blood types are different between mother and child, and this blood gets mixed during the delivery of the baby. So for the second pregnancy, there's a significant issue then with the adverse reaction of these bloods mixing. So that completes our review of the blood chapter, and in that way it also completes our review of the entirety of the study guide for this chapter. I'm not uploading the completed version. Like I said, these completed versions are only available through these lecture videos or review session videos or by attending to a review session. So I recommend watching those if you have any trouble filling in the study guide. And I think that we're going to jump back to our initial PowerPoint. So bear with me as we move back there. And the very last thing that I have left to say in terms of this review session preparing you for the exam is that if you do feel that you would benefit from having tutoring, remember the college offers tutoring through the Cranium Cafe. I've already discussed it in the past. Remember to look up the link if you want to do that. And in addition to that, I'm of course available for one-on-one group, small group, or the larger entire class tutoring sessions. So just let me know if you feel that you need help. At this point, I would normally entertain any questions, but since this is a video, I'm just gonna leave it at that. Feel free to message me directly. And I think that wraps up our session for today in terms of reviewing what might come up in the exam. I thank you for joining me, and I hope you have a very good week. I hope you find that actually the exams are not too bad. I think that they are definitely worth of taking. It gives you and it gives me a chance to see how are we doing with covering the materials. Remember, all of my exams are open book. So you can use your notes, you can use your textbook. I really encourage taking good notes. Remember, however, that these are timed. So there is a reason why there is a time limit. That means that you don't really have a chance to look up everything of every question. So you do need to know the material. And the final thing that I do highlight is that copying exam questions or sharing them would be a problem. They are not to be shared. That's why I am doing open book exam. If I find that happens, then we have to go to proctored exam format. Like I said, for the practical, make sure you practice. There's no new question, no new pictures or curveballs, but the big difference is that you have to be comfortable with typing in the answer. For both the exam and the practical, I will be doing manual grading, so your initial grade will not be your final grade for that test or that element of the test. Normally the grades go up a little bit once I do finish the final manual grading. So I recommend you not to get too upset or too worried if it's not exactly where you want to be with the grade, and wait until you hear from me that your grades have been finalized. As always, reach out to me if you have any questions. If you feel that any part of the material feels challenging, I hope you have very good week and I wish you best of luck with the test and practical. And finally, remember, if you need extension, if you need to finish off any assignments that you might have missed, reach out to me with details of what assignment are we talking. On that word, have a very good week, and I look forward to seeing you acing the exam.